All right. Hello, my name is Billy, and I will be teaching IB Bio HL this winter. And today I'm just doing a demo class of about 10 minutes on nucleic acids. And this is the topic that we'll be starting with when we start our sessions in later this year. So what I have figured is that you probably already have done, hopefully, DNA units on DNA, which are units, I believe, 3. And from there onwards, the SL level covers what DNA really is about and the basic structures. And now that we are in HL, we are going to be covering more in detail of how nucleic acids behave and what happens to the nucleic acid using a lot of different enzymes to be replicated, to be transcripted, and also to be translated into proteins. So today's class, or this demo class, will be covering just the basic DNA structures, but in a lot more depth. And every class we will start by going over the syllabus. So today we'll be learning more about the DNA structure, as I said, and then really going deeper into how the carbons are actually important in the deoxyribose, and then going from there onwards, how that will be applying to furthering our understanding of the DNA replication process. So before we begin, we do have page 88, which basically covers how DNA was found. There were a lot of researchers that got involved back in 1950s of how to understand our DNA structure. They knew that DNAs existed or genes existed, but they really wanted to know how they were configured. So there is some reading that you can do on here, and I'm going to have you read this as homework. So we'll be covering this topic next class. So let's go into DNA structure. So for the most part, you probably already know that DNA stands for deoxyribose nucleic acid. So what is a deoxyribose? Well, it looks like this, right? Unlike the other kinds of sugars that we are already well aware of, such as glucose, it's in a pentagonal shape, and it has an oxygen in the middle, which is very important to understand because that's what makes the pentagon. And then the fifth carbon sticks out of the ring. And this goes the same for deoxyribose as well as the ribose. The only reason between the two naming differences is that the carbon here does not have an oxygen attached to it, whereas in the ribose, we have oxygen attached to the carbon here. So let's go understand what this deoxyribose really does in a DNA structure. So we know that we basically usually draw a nucleotide like this. We start with a sugar, and then we say that it is attached to a nitrogenous base, right? And nitrogenous bases can be adenine, thymine, guanine, and cytosine, right? And we'll be talking more about this a little later on. And then you probably have seen something that sticks out like this. And this is the basic structure that we usually use when we are teaching on a SL level. And we're going to dissect this a little more. In that, well, let me redraw the pentagonal shape. And I said the carbon sticks out like this, right? So this is the basic deoxyribose structure. And what we have to first know is that we need to learn that the carbons here are all different from one another. This carbon is different from this, and this. Thus, chemically, since they're different, we have to number them as 1, 2, 3, 4, and 5. For some of you who are already doing chemistry, this might be familiar, but for those who are not, just know that it goes clockwise. It doesn't have to always go clockwise, but because the nitrogenous base is attached to the carbon over here, this becomes the number 1 carbon from there onwards. So always remember that to the right side, the carbon over here, the carbon number 1 has a nitrogenous base, or the carbon that nitrogenous bases are attached to are the carbon number 1. And from there onwards, we're going to name them consequently. So carbon number 1 is important to know. And then the next one over that is important to note are 3 and 5. These are very important because these two carbons are the carbons that are used to link up the free nucleotides here. And actually, I didn't draw one part that is very important to the nucleotide. Oops. Some of you may know, some of you may not. Is that this carbon actually is attached to a phosphate, which is PO4. And this PO4, this phosphate group, is the one that actually bridges between the nucleotides. And this number 5 has phosphate attached to it. And then, so if we have this nucleotide, we're going to have another one that comes right below it. If we were to make a whole chain of DNAs, and then we are going to have that phosphate group attached to the carbon 3. So the C5 is where PO4 is attached. And C3 is where PO4, oops, is received. Does that make sense? So since the DNA attachment from one nucleotide to another nucleotide goes from C5 to C3 via a phosphate group, we always have to remember that any kind of DNA nucleotides getting attached to one another goes from 5 to 3. Always have to remember. And it's sometimes very tricky because obviously 3 is a smaller number. So you would always think perhaps that 3 goes to 5. But always remember it goes from the 5 carbon to the 3 carbon. 5 carbon of the nucleotide where that it's going to attach into the next nucleotide into the 3. So it's 5 of the prior nucleotide to the 3 of the next nucleotide, right? So here we have to remember that when we say it's 5 to 3, it's 5 of the prior nucleotide to the 3 of the next nucleotide. So if we were to keep on drawing this out, we're going to end up with a chain of 5 to 3, of 5 to 3 on one strand, right? Then this is the DNA, so it's two-stranded, right? So we have to draw the other strand. How would that be drawn? It's actually a mirror image. But in a more distorted manner. Actually, I'm think I'll take that back. It's not a mirror image. It's more of a flipped backwards. Where the nucleotide will be facing each other, but the oxygen comes below. And then here, since this will be the number 1 carbon, it's going to be 2, 3, 4, and then the 5 carbon that sticks out. And then it's going to attach into the next carbon. Next nucleotide is carbon 3. And then the same goes with the 5 going into the 3 carbon. So do you see how the progression of the 5 to 3 here goes from this direction on the left side, whereas on the other side, it goes to this direction, right? So we're going to form 5 to 3, whereas it's going from 5 to 3 in this direction. So what we find is that it's parallel. However, it's going the other way, the opposite direction. So opposite meaning anti. So we call this structure an anti-parallel structure. So just based on the deoxyribose having 5 carbons, named from 1 to 5 this way, and then the 5th carbon, which sticks out from that ring, having a phosphate attached to it, which is used to bridge the nucleotide into the next nucleotide to the 3rd carbon of the next nucleotide. And because this flows from a 5 to 3 fashion, the DNA structure, the double-stranded DNA structure has a anti-parallel structure. So everything gets tied together, and that's how you should be remembering the DNA structure.